really appreciate the opportunity, and I'm truly honored to be here and have the opportunity to present. I wish I was there in person. And thank you, Jonathan, for the invitation. Just sharing my slides. Hopefully everyone can see that. I do have a few disclosures, so I'll show those here. I have received grant support from United Therapeutics and its subsidiaries, and I will be discussing the investigational use of both the U-kidney and the U-thymo kidney. I think the other major disclosure to make is that I am, in fact, a transplant surgeon and not a nephrologist. So you can imagine in my world, nothing more intimidating than giving a talk to a group largely comprised of nephrologists and patients. But thank you again for this tremendous honor. So as everyone in this room and audience knows, really end-stage kidney disease is a huge problem in the United States. If we back it up further, we know that there are 37 million Americans who have chronic kidney disease, many of whom will see progression of their disease and actually develop end-stage kidney disease. And there are hundreds of thousands of Americans with end-stage kidney disease, somewhere around 700,000 to 800,000 currently. And what we know is that there is, in fact, a cure for end-stage kidney disease, and it's in the form of kidney transplantation. And kidney transplantation has really been established as the gold standard therapy. And this is a graph produced by Dr. Wolf and colleagues. And you can see on the y-axis is the relative risk of death, and then on the bottom x-axis is days since transplantation. Just to orient folks, the dotted line here is your risk of mortality if you stay on dialysis. And so you can see immediately after the transplant surgery, your risk goes up, and that's common to any surgery that you might have. But you can see by a little over 100 days after your transplant, that line crosses and goes below that dotted line. And that's really where you can see that transplantation begins to offer a significant survival benefit over remaining on dialysis. And so really, our goal would be able to offer a kidney transplant to anyone in need. But we know, in fact, in this country and really around the world that there is, in fact, an organ shortage. You can see here, most recent numbers have us around 80 to 90,000 people waiting for a kidney transplant. And we do somewhere between 20 and 25,000 per year. And I think this 80,000 to 90,000 number, it really bothers me because that just represents a fraction of those patients who actually have end-stage kidney disease. So we know the majority of patients who have ESKD never even make it to the transplant waiting list. And of those who make it to the waiting list, very few actually get a transplant. And you can see these bar graphs in the bottom, the blue being deceased and the red being living donor transplants, that the curve has really remained flat for the last 20 years. We know that about 40% of waitlisted patients will be dead in 5 years. And we know that therefore the organ shortage really is lethal, and it is a refractory, severe, and impactful problem that needs a radical solution. And we would certainly, the government also has acknowledged this. The first ever executive order on a chronic disease came out in 2019 on advancing American kidney health. And they really encourage the development of the artificial kidney. And we would certainly argue that xenotransplant could be viewed as sort of an artificial kidney. And really, perhaps it is an impossible solution that perhaps we can make possible. And I do want to thank Dr. Rothblatt. She is the chairperson of United Therapeutics. And she really has been very involved in wanting to make xenotransplantation a reality. For just a brief background, she actually made her fortune initially by developing Sirius XM satellite radio, but became very interested in healthcare when her daughter was born with pulmonary hypertension. And Martine's goal has really been to be able to have an organ on a shelf in the event that her daughter ever needs a lung transplant. And that's really how she became involved in xenotransplantation. And this is just a really great quote from her, that when she sees the word impossible, she drives and acts right between the M and the P. And she says, no, I'm possible, or anything's possible. And I think that's really been her story in this field, and really the field of xenotransplantation. So if we back this up, and what is this concept of xenotransplantation? But essentially it's the concept of taking an organ from an animal and being able to use that ideally in a human. And certainly we have settled on the pig as the ideal animal source. So why might that be? And when I first got into xenotransplantation, I will be honest, I really had to educate myself on all of these things. But the reason a pig is an ideal animal source is that they can reproduce quickly. So pigs have about 2 to 3 pregnancies per year. They typically have large litters, about 12 piglets per pregnancy. They grow very quickly, so they're only about a kilogram at birth, but by 6 months, they're already up to 130 kilograms. And they have a long lifespan of about 30 years. We also know that their kidney function is quite similar to humans, and that's what that table below shows. So you can see the various parameters, human here and swine there. I'll orient you to the glomerular filtration rate, which I think most people are familiar with. You can see for a human, it's about 130 mils per minute, and in an average swine, it's about 126 to 175. So that's all great, but we know a pig kidney in and of itself will not work in a human because the human body would recognize the pig kidney as foreign and reject it. So what then to do? And this is really where genetic editing has come into place, where we're really genetically modifying or sort of humanizing the pig kidney. And really the goals, I think, have been to overcome immunologic incompatibility as well as viral transmission. And as a field, xenotransplantation has been around for a very long time. But recently, with the introduction of CRISPR-Cas, and I think it's important to acknowledge Dr. Charpentier and Doudna, who won the Nobel Prize in Chemistry in 2020 for this, that really increased the precision as well as how quickly genetic editing could occur. And via high throughput engineering, we've really been able to genetically engineer human compatibility, as well as in some cases actually eradicate retroviruses from the pig's genome. And this is really, I think, why you're seeing this resurgence of xenotransplantation in the last year in particular. So one of the things that we know is the genetic edit or gene editing has really led to the success of pig to non-human primate xenotransplantation. So that's really the model that we primarily tested these genetic edits, where you take a kidney from a genetically edited pig and you take a non-human primate, typically a baboon, you remove the baboon's native kidneys, and then you transplant this genetically edited kidney into the baboon. And what we've learned is that we have observed life-sustaining renal function in these non-human primates. We've been able to see urine output and creatinine clearance, as well as electrolyte homeostasis. The graphs below are just some graphics of 10 gene-edited pig kidneys into non-human primates, showing serum creatinine as well as electrolytes like potassium, phosphorus, and then protein like albumin. But what we also know is that our genetic edits have really been designed to improve compatibility for humans, not non-human primates. So to orient everyone, the arrow points to something called a negative cross-match. And a cross-match is something that we do in transplantation to ensure that there will be a tissue match or compatibility between the donor organ and the intended recipient. So if you look at the boxes up at top, the arrow points to this little small lower left-hand box. Any sort of dot, if you will, that falls into that would represent a negative cross-match, meaning there would be a good tissue match and the organ would be unlikely to be rejected. So this top series is looking at humans, and it's testing it against a wild type pig, a pig that has had a single knockout of one of the carbohydrate antigens, a double knockout, and then a triple knockout. And what you can see is that as you progress from left to right, you see increase in compatibility, such that by the time you get to the triple knockout, you actually see that about a third of humans have a negative cross-match with a triple knockout genetically edited pig kidney, compared to none if you just look at a wild type kidney. If you then look here at non-human primates, what you'll find is that really no non-human primate has a negative cross-match with these genetically edited kidneys. And the graphics at the bottom show the same thing. So this is again looking at wild type and then going all the way to triple knockout. You can see that humans have negative cross matches here with the triple knockouts. Anything below this dotted line is a negative cross match, whereas non-human primates, including baboons and rhesus monkeys, never achieve that. And what we know is that really and truly the pig to non-human primate kidney xenotransplantation model has really been a model of incompatible kidney transplantation. And unlike in humans, in non-human primates, it's been very difficult to do any pre-transplant immune modulation in terms of trying to remove these antibodies. And so really you're trying to go across a positive cross match. And that's really what these data show. So on the left-hand side, This is looking at 183 non-human primate sera. And this has been tested against triple knockout pig cells. And you can see that the frequency of a positive cross-match was 100%. And even among those animals that had low titer or low level cross matches, if you look at the graft survival, you can see that it's quite poor. It's less than 25% at 100 days. And so that's certainly not something that we would want to offer to a human if it's truly got graft survival that poor at 100 days. But again, these pigs have been genetically edited to optimize them for the use for a human, not a non-human primate. So really there are critical knowledge gaps, and although we have genetically engineered these pigs such that their tissues look less foreign to the human immune system, and in our case we work with a 10 gene edited pig that consists of that triple knockout background that I described, which are the 3 main carbohydrate antigens thought to cause hyperacute rejection. We've also knocked out the growth hormone receptor of the pig kidney, and the goal there is to make sure that the kidney does not outgrow its new host. And then we've also inserted 6 transgenes to try to enhance compatibility. And while we've tested these in non-human primates, we know that non-human primates are immunologically different than humans. And we also know that we need to be able to test this with conventional immunosuppression. The immunosuppression that's largely been used in non-human primate models is not FDA approved and is not immunosuppression that we typically use in human-to-human allotransplantation. And so we've had to do that because of that incompatibility, but we certainly would rather not do that in human transplantation. So what then were we to do to try to answer these critical knowledge gaps? And what we realized is that we really needed a human model to be able to test our genetic edits, but we needed a human model that wouldn't actually risk a living person. And this is where we began to think about the concept of human brain death as a novel preclinical human model. And this is actually an idea that Tom Starzl, the father of liver transplantation, actually sort of began discussing many, many years ago. And so we thought perhaps this could help us. And so the design advantages would be that we would not harm a living person, as this human being would have in fact been declared brain dead. We'd be able to test our engineering in a human before the living, and we'd also be able to test our immunosuppression. And so we worked very closely with our organ procurement organization known as the Legacy of Hope to design this model. So we were able to identify a brain dead human who was not able to donate their organs for the purpose of transplantation, but was interested in participating in research, and their family agreed to that. And then perform this Xenotransplant in that context and follow that human brain dead decedent for a period of time. But before we could do that, we also, as you think about trying to move this into humans, whether brain dead or the living, we also have to be able to ensure that we don't have transmission of a disease or a zoonosis, so diseases that are specific to animals and not humans. And so in order to do that, we really needed to establish and develop a pathogen-free facility. And this is a journey that UAB began back in 2016, in which we developed a pathogen-free facility that's about 30 miles from campus. And this is where these genetically edited pigs are born and bred, and they're behind a barrier so that we know exactly their pathogen status, and we can ensure that they don't have any diseases that we would not want to transmit to a human. It also allows us to confirm our source animal pathogen status prior to procurement of the kidneys and prior to transplantation. The other thing that we needed to do is really ensure that the pig kidney was appropriately sized. I mentioned that we're working with a 10 gene edited pig that includes the knockout of the growth hormone receptor. We added this knockout because without it, we noted that the pig kidneys would continue to grow in the human, or in the host, in this case, in a non-human primate. And we certainly didn't want that to happen in a living person. So with the knockout of the growth hormone receptor, you really see that the kidney growth is no longer exponential. You can see that it plateaus. You can see that in the blue and green lines. And then in this upper left-hand corner, this is the typical dimensions of a human kidney and how much a human kidney typically weighs. And so we were able to take serial ultrasound measurements looking at the pig kidney's width and length, as well as pig kidney weights at the time of procurement. And we were able to determine that a 10 gene edited pig at about 180 to 200 days of age would have a kidney weight of about 160 grams. So now that we had this sort of preclinical human model, we had a pathogen-free facility so that we can ensure that we wouldn't have disease transmission, as well as being able to ensure the appropriate size of the xenograft, we felt that we were ready to move forward. We had a huge study team. This resulted in a lot of planning over about a 2 to 3 year period. It included everyone from our sponsor, United Therapeutics, to our IRB, to our animal version of an IRB, IACUC, as well as external ethics review. But on September 30th of last year, we performed the first clinical grade, meaning the kidney came from a pig that was in a pathogen-free facility, kidney xenotransplantation into a human being. And we used human brain death as the preclinical human model. And definitely want to shout out to all of my co-authors, in particular, the first author, Dr. Porritt. And we learned quite a number of things from this. So with the first clinical grade 10 gene edited pig kidney into a preclinical human model, we were really able to establish sort of the whole transplantation process itself. So we were able to have the procurement facility at our pathogen-free facility. We built a human clinical grade operating room behind the barrier. So the procurement happened there. The pig kidneys were removed, they were flushed, and they were packaged in a standard fashion, just like kidneys are removed in the setting of brain death for the purpose of human-to-human transplantation. You can see Dr. Arandi, one of our transplant surgeons, and Sarah Macedon, one of our nurses, coming back from the pathogen-free facility. The pig kidneys are packaged and in this styrofoam cooler, just like human kidneys are. This is the human decedent. So you are looking here at the native vena cava. It's just showing you that the right kidney has been removed. This is the undersurface of the liver and gallbladder. This picture here is showing you what the pig kidney looks like prior to reperfusing or establishing blood flow to it. You can see that it's pale and lifeless, and that looks just like a human kidney looks prior to transplantation. And then you can see this beautiful pink kidney, and this is after we have established blood flow to the kidney. This is a Doppler probe where we're listening to signal through it, and it sounded beautiful. And within about 20 minutes of reperfusion, the pig kidney inside this human, recently deceased human, began to make urine. And I can honestly tell you, it's probably one of the coolest things I've ever been a part of, and was just truly remarkable. But the other thing that this study really allowed us to do is establish a cross match. And I want to orient everyone, but this is a standard of care, and it is mandated by the OPTN in the setting of human-to-human allotransplantation. And you can see here the policy 4 and 4.6 is cross-matching. And really, it's a requirement to perform a final cross-match and report the results to the transplant program before transplant. And the reason for this is because if the cross-match is positive and you go across that without intervening, something called hyperacute rejection will occur, and the kidney, when you restore blood flow to it, it initially turns pink, and then within about a minute to 2 minutes, it begins to clot from the inside out, and it turns completely black, as it's shown here in this picture. And that is what's called hyperacute rejection, occurring minutes after reperfusion, and is related to high levels of donor-specific antibody. And certainly that would make a transplant futile. And that is why we are required to have a cross-match prior to going to transplant. However, in the setting of pig xenotransplant into human, no such cross-match existed. So we had to develop this, and we developed the first flow cross-match for human use in the setting of xenotransplantation. So this is showing you a prospective cross-match, meaning we did it prior to the transplant. This is our negative control, positive control, and then this is our human decedent. And you can see that we had a negative, this is the negative control, and then you can see the shift for positive. And then the decedent here aligns with the negative control, meaning that we predicted ahead of time that there would be a negative cross match and that we would not see hyperacute rejection. However, the only way to validate this was to actually leverage this preclinical human model that we have labeled the Parsons model after Mr. Parsons, whose family enrolled him in this study after he was declared brain dead. And you can see pre-reperfusion and again post-reperfusion that the kidney did not turn black, And so there was no hyperacute rejection. And this really allowed us to validate the cross-match. And this is really filling a major knowledge gap when you look at moving pig to human kidney xenotransplantation into the living humans. The other thing is that we know that standard of care in human-to-human allotransplantation also requires infectious disease testing. And this is deceased donor organ procurement policy number 2. And you can see that this policy requires deceased donor infectious disease testing on a number of different things. And what's really important is that we require both PCR-based assays, so looking for active infection with certain viruses, as well as serologic assays looking for prior to exposure to certain viruses, particularly for viruses that can be dormant or latent. So this becomes really important when you think about diseases that can come from a pig and be transmitted to a human. How do we ensure that that doesn't happen and have standards that mimic human-to-human allotransplantation? So in our study of the first clinical grade porcine 10 gene edited pig kidney to a human, we were able to demonstrate no pig to human disease transmission. So pig positive is the wild type pig. Pig negative was our 10 gene edited pig. NC is normal control, and then day 0 through day 3 represents blood from the recently deceased human that was enrolled. PERV is porcine endogenous retrovirus. It is PERV-C that is known to infect human cells and what we do not want to transmit. You can see that the wild type pig had PERV-A and B as well as PERV-C. The 10 gene edited pig had PERV-A and B but did not have PERV-C. And what is important is that our human recipient, our human recently deceased human recipient, did not have any PERV transmission in their blood on days 0 through 3 of PERV-A and B or PERV-C. RPL4 is a ribosomal protein that's unique to pigs, and you can see that both the wild type and our 10 gene edited pig had this, but our human decedent did not, and that means that there were not pig cells floating in circulation, meaning no chimerism, which is another important finding. But what we do know is that despite us demonstrating that we did not see disease transmission of a porcine endogenous retrovirus, we do know that the gaps do persist. Many of you may be familiar with the story in the news from earlier this year of a living human who received a heart from a 10 gene edited pig. And one of the things that we learned from this case is that porcine CMV or porcine cytomegalovirus DNA was in fact detected in the first pig to human heart xenotransplant. We also know that they did confirm the source animal pathogen status prior to procurement, but that this was done only via PCR, meaning they were only able to detect active infection, and the pig was negative for active infection. But we also know that the source animal was from a biosecure facility, not from a pathogen-free facility. And this is very important because it may mean that this particular animal actually had been previously exposed to porcine CMV, which is a latent virus. So while the animal did not have active infection, it may have been exposed, and latent viruses can sort of come back. So viral latency is a type of persistent viral infection, which after initial infection, viral proliferation ceases, but the genome is not eradicated. So they can then reactivate and produce viremia, particularly in a host that is immunosuppressed. We see this in human-to-human allotransplantation. Patients often will get CMV, but porcine CMV is something that we definitely do not want to transmit to a living person, as it can be associated with complications, including a coagulopathy. And so we know we can eliminate porcine CMV through herd elimination, and that's part of why we have our pathogen-free facility. But what we also know is that up until very recently, there was no serologic test to be able to examine animals for past exposure. And our group has certainly worked now to develop this, as we see that as something that's going to be necessary prior to moving this into living persons. The other knowledge gaps that I think persist is that we don't know if there's really creatinine clearance after pig to human kidney xenotransplant. So there have really been 3 cases reported in the literature using the preclinical human model that I described. One was our study. We removed the native kidneys from the brain dead recipient and then did our transplant. And while we saw urine output, we did not see creatinine clearance. In the other case series, the native kidneys were actually left in place, and the authors really did not examine or isolate the xenograft function from the human kidneys. So there was no way to attribute the creatinine clearance to the xenograft itself. And so we still don't know what the creatinine clearance after pig to human kidney xenotransplantation will actually be. We also don't completely understand some of the histology we've seen. And so I think the etiology of histologic changes is going to be really important. So in our particular case, we did not see any evidence of C4D deposition, which is typically observed in antibody-mediated rejection. However, in the other cases reported, they did see C4D deposition, and I think additional work is going to be needed to really understand the histologic changes so that we understand how to account for this and potentially avoid early antibody-mediated processes when we move this into living persons. And then I think the other knowledge gap persists, or what are the exact ideal genetic edits? In the animal that we used, we had 10 gene edits. In the report by Montgomery and colleagues, they just had the alpha-gal knockout, although this wasn't reported that in the New England Journal paper. This was reported in the lay press. And so I think at the very least, we can say that we need the alpha-gal knockout. I think some of the in vitro data that I showed earlier, however, would suggest that the triple knockout of all the carbohydrate antigens are actually probably really important. So what are conclusions and next steps? And I know I threw a lot at you in a very short period of time and look forward and want to make sure we have time for questions. But I think the knowledge gaps that we have filled by moving this into a preclinical human model, really, we know that we can avoid hyperacute rejection in humans. We know that the alpha-gal knockout is, in fact, a critical gene deletion. I think what knowledge gaps persist is what type of function will we see in a living human? In other words, will these genetically edited pig xenografts be a bridge, meaning they bridge the patient, having them avoid dialysis for several years until they can achieve transplant with a human allograft? Or will they actually be destination therapy, meaning you get a kidney from a genetically edited pig and it lasts you your lifetime? So that gets at the durability of transplant. I think we also need to further examine predictive capabilities of different cross-match techniques. So I showed you a flow cross-match, but we know in human-to-human allotransplantation, we also are able to run things called single antigen beads that really allow us to understand antibody specificity. That currently does not exist for xenotransplantation. We also need to understand what our optimal immunosuppression regimen is. So in the case that we did, we use standard immunosuppression that's typically used in human-to-human allo. And we know that we did not see hyperacute rejection in that context, but whether or not that's going to need to be tweaked, I think, is yet to be seen. The gene edits, do we have what's necessary? Is it sufficient? Are we going to need to make tweaks to that? I think the molecular mechanisms of graft damage in human recipients is going to need to be better understood, as well as potential disease transmission, how to avoid it, and what to do if it happens. And then I think one of the other things that's going to be really important is if you receive a kidney from a genetically edited pig, is that somehow going to sensitize you such that it makes it harder for you to get another transplant in the future from a human? Our early data suggests that we don't think that will be the case, but the only way to definitively test that is to move it into living persons. And I think, how do all of these studies inform next steps? I think certainly my colleagues and I think that we do need to begin thinking about moving this into phase 1 studies of safety and efficacy in living persons, but that the trial design probably needs to be adaptive such that you're learning from each transplant. So with that, I do want to thank all of my colleagues. This is a picture of just a small fraction of all the people who've been involved in the Xeno work at UAB. I want to thank the School of Medicine as well as UAB Medicine and our organ procurement organization, Legacy of Hope. So thank you very much, and I look forward to answering your questions.
Well, thanks, Jayme, for a fabulous talk, really tour de force, and for incredible amount of work that it took persistently behind the scenes to get to the point where you could do this first, if we want to call it a human model experiment. That was really tremendous. Let me see if there are questions from our audience for you. And do we have a microphone for the audience? Microphones are in the corners if anyone wants to come up.
Thank you, Dr. Locke. Really appreciate the talk. It was terrific. My name is Larry Kessler. I have one question for you, and it's about international collaboration. One of the things that we've tried to model in ideas is to try to build international collaborations, because we feel like no single institution can solve all these problems. It sounds like you have massive problems to solve, but you're making a lot of progress. Are you able to collaborate well internationally? What forces are being brought together outside the United States, or even across collaborators in the United States, that can actually make this advance in as reasonable and quickly a way as possible, given that the struggles are going to be really intense?
Thank you so much. I think that's a really important question. And so one of the things that has happened, so the American Society of Transplantation, as well as the American Society of Transplant Surgeons, has actually put together a task force that's being led by Dr. Alan Kirk. And it's really bringing investigators together, both from the US as well as international, to put together sort of this idea and notion of having sort of a registry. There's a lot of interest in that. We also were able to present, the FDA had an open public hearing, and Dr. Kirk was able to present some of that work and ideas to the FDA during that hearing. And one of our recommendations was to have a sort of registry where we can actually share these types of data and be collaborative. I think in many ways the field has been a bit siloed. And you can imagine, there's a huge amount of investment from these various companies in this. And so intellectual property becomes an issue and other things. But certainly, I think there are things that we can share that are going to be absolutely critical and vital to be able to move this forward quickly. And that's really what our patients need. And so I will tell you, there's great interest in the scientific community to have this be as collaborative as possible.
Thank you.
Dr. Locke, let me also say that was really an extremely informative and excellent presentation. Thank you so much. Maybe I missed it. How many days did this brain dead model where the individual was alive? And were there any other physiologic consequences that were monitored? Spasms and things like that you might have seen over that period?
Thank you for your question. I don't think I described that. So the individual was deceased as they met brain death criteria, but the body itself was, blood flowing through it, heart beating, all of that, just like a typical deceased donor. And so our commitment is to have every deceased donor be able to donate an organ if at all possible. So it took 5 days going through that process before it was determined that that was not going to be possible. So on day 5 of brain death is when Mr. Parsons was enrolled in the study. And then the study went on for about 77 hours after that. And that limitation really reflects brain death. Brain death is a very challenging sort of environment to have stability. We did not note any sort of instability related to the transplant itself. Going into it, he was already in extremis on multiple pressors. as well as in something that we call DIC. So it was a sort of a tour de force to even be able to do this and have the study last 3 days. And I think that's really one of the limitations of the preclinical human model, is being able to have that go on for a long enough period of time to be able to test some of these later endpoints. I think it's also really important to note that even if we didn't see issues related to brain death in terms of stability, this does interrupt the family's bereavement process, and they have to be prepared for that because we're holding on to their loved one for a prolonged period of time. It's part of why we really engaged our ethics colleagues as well as our IRB. Because the individual's brain dead, technically it does not meet human subjects research, but we wanted it to be under that umbrella so that the family could be fully informed of what was going on. And so I think as we look at this preclinical human model, I think these are the things we're going to have to keep in mind when we start wanting to have longer endpoints. I'm not sure we're going to be able to achieve that. And we're at some point, I think, going to have to move this into living persons to get some of these questions answered. The question is, do we have enough safety information to feel comfortable doing that now, or is there more work that needs to be done?
Okay, thank you. It is very complicated.
It is,
Appreciate your efforts there.
Thank you.
We have a few questions coming in through Zoom chat. The first one is asking, do pigs have different blood types like humans?
So they do, but they've been genetically edited to essentially be a universal blood donor. So in this case, all of the pigs are blood type O.
And another question, while we're in the learning phases of renal xenotransplantation, where the stakes are very high, how would you describe the characteristics of the ideal first humans to receive a renal xenotransplant?
It's a great question. I think it's something that the community continues to debate. I think it will largely be driven by what the FDA thinks. I think it's really important that we not do anything that is futile. So I really think this should be done in the setting of a clinical trial, a phase 1 trial, as opposed to emergency authorization uses. I think it's harder to learn in those settings. I also think it needs to be in someone who meets criteria for transplantation. In other words, they have to be compliant, they have to have reasonable function, those kinds of things, because we need to be able to follow this individual for individuals for a prolonged period of time. Even if the xenograft fails, we need to be able to follow them to ensure that there are no disease transmissions or other things that occur later down the line. And so with that, I think that it really is a risk benefit. So I would sort of suggest perhaps it be someone who does not have a living donor, who is relatively new to dialysis, who has a very long waiting time, and who may be interested in helping drive this very innovative idea. But I think it's going to take a very special series of individuals wanting to participate.
Another question virtually. Dr. Locke, really amazing progress. How do you think about the challenges faced by decellularization or resellularization method of xeno organ development versus the gene-edited method you had outlined?
I think it's a great question, and certainly I think Dr. Rothblatt does as well. I think this is going to take a multi-pronged approach to have artificial organs available. So I think you're seeing processes moving simultaneously, and hopefully we'll have both. I think certainly the genetically edited model that we have now suggests about somewhere between 1/3 and 1/2 of the humans that we've tested, human sera that we've tested, are actually compatible, but that still leaves a pretty big gap that I don't think allo transplant or human to human is going to be able to fill. So I think that's certainly an option. And just kind of going back to the last question about the ideal person, I also think a highly sensitized patient that doesn't get a lot of offers, so that sort of CPRA of 100%. And the reason I say that is because for whatever reason, your CPRA does not seem to matter with these pig kidneys. So we have multiple high PRA human sera that have a completely negative cross-match with these pig kidneys. And so I think that really affords a group of folks who might otherwise struggle to get transplanted an opportunity to come off dialysis.
And the final question from our Zoom viewers. Thank you, Dr. Locke. Do we have enough information to know if people who have had a human transplant would be eligible for a xenotransplant?
Great question. I think I kind of just alluded to that. So highly sensitized patients are often that way because of previous transplants. That does not seem to preclude receiving a pig kidney, meaning your level of sensitization does not predict the cross-match compatibility with the pig. They seem to be completely independent of each other. So I think that's absolutely a possibility.
Good morning, Dr. Locke. My name is Eric Ditchman. I'm a 21-year patient on dialysis, mostly home, but I've had 2 transplants, one from my wife and one from a deceased donor. I have 2 questions. One is, how has patient-centered outcome research principles been involved with your research? And secondly, would we be able to knock out protein receptors for like focal segmental glomerular sclerosis that reoccurs? Right? That's sort of more personal. My larger question is about the patient-centered outcome research.
Both fabulous questions. There are so another exciting possibility is there really no data to suggest that something like FSGS will actually recur in the pig kidney. So this may be a great option for patients who have diseases that are known to recur and have already had disease recurrence. So that is a huge issue. And then in terms of patient-reported outcomes, that absolutely has been discussed, and I think must be a part of early phase 1 trials. I can tell you we have done here at UAB some formative research in the community trying to understand patient perceptions around xenotransplantation and making sure that we are approaching this in a very patient-centric way. And I think that's going to be really important to move this forward because without question, our patients are going to be our partners. They are a critical component to this team to really make this a reality.
Thank you.
Time for one last question.
Before I begin, I just want to say this was really amazing work. I really commend you. I think this is a really important topic, and you've made tremendous progress. My question is kind of just about, you mentioned that when you're going through this, there was some issues in terms of creatinine clearance. You didn't really see much creatinine clearance. I guess, what are kind of the steps you see going forward to maybe address this issue? Do you think they will still need to go through dialysis after the transplantation? Just wanted to get your thoughts on this.
No, great question. Ideally, we would be able to identify a brain dead individual that we could enroll very early on, meaning we know sort of from the outset that they're not going to be a candidate to donate their organs for transplantation, where we could actually do the xenotransplant early on in the brain death process so that we're not dealing with all of the sort of instability and all the other things. I mean, I'm not at all surprised that we saw delayed function, at all. The Mr. Parsons already had a pretty significant acute kidney injury going into all of this. So he was already quite unstable. So I think that is one thing that we can certainly do to address that. I think that there is going to be the possibility of delayed graft function or even primary non-function. And so I do think we have to be prepared that the first living human recipient may need dialysis immediately post-transplant. Our goal would be to not have that happen, because it is, it would be scheduled. So the idea is to have this transpire similar to a living donor transplant. So it's scheduled, it's happening. And so the cold ischemia time that the kidney experiences will be very short. So our hope is that we won't see that, but I really think that the only way we're really going to know that is as we move it into living persons.
And thank you, and thank you, and we really enjoyed the presentation. It was really awesome.
Well, thank you so much. Like I said, it's pretty intimidating giving a talk to a group of nephrologists and patients, and so thank you so much for having me. I really appreciate it.
Thank you so much, Dr. Locke. That was a great talk, and good luck with all your work in the future. It's really tremendously important work.
Thank you.